Welcome, this is the Help Me video for Key Assessment 3 Core Concepts Paper 3. Meselson and Stahl investigated DNA replication, and they tell you a little bit about the experiment. Your eyes should be lighting up seeing this and go, right, I know about this. Pretty straightforward question. Identify 3 variables which would need to be controlled. So variables controlled, that's a controlled variable. So things that you are keeping the same. So the things that we are changing are the generations of the bacteria, but things that we are keeping the same in nearly every experiment. Think about temperature, I will give you that one. You only need 2 of them. So temperature, and try and think of something else. So what they're doing here, they are spinning these tubes. You can think they're adding things to the tube beforehand. So what would you make sure you did if you added something to a tube and spun it round? And no, actually you need 3, don't you? So 3 for 2 marks. So 1, 2, 3. Then we've got this experiment, the classic experiment, and the only little trick that they've done is that the dense DNA is C, whereas the light DNA is A. Yeah. So that's the bit that you need to interpret, because at the start the bacteria were cultured in only the 15N. So that's the starting one. So at the start there you'd expect 15N, and that's going to be our C. And the lighter DNA is going to be A. So as long as you get those the right way round, and then you're into identifying A, B and C now. So that's at the start you've got the heavy, and then you can see the peak there for C. And one of them will be in between, so A, B and C. So A and C are the 2 forms of nitrogen, and the one in the middle is the hybrid. So you just need to put that into numbers with ends. And then part 2 and 3, 7 marks. If you know the tail of this experiment, you'll get 7 marks. So it's really an essay. It's really an essay, is this, but they've split it up into 2 parts. So generation 0 and 1. So 0, we are all heavy. Generation 1, well, the band has moved up and the peak has moved across. So you just need to say that it's a mixture of the 2 types of DNA. And why? Well, semiconservative replication. I'm going to write what I know about semiconservative replication here. I'm going to say about the DNA splitting and each strand of DNA becoming a template for the new strand, and the old strand is going to be the heavy DNA and the new strand is going to be the light DNA, and so it's going to be a hybrid or an intermediate. So you just really need to put that into words, and then later on, just pretty straightforward again. You are starting with the intermediate. So at the start we've got all heavy. Then generation 1, we've got the intermediate. So we'll get 2 strands of that. And then in generation 2, these are going to split up again. So we split these up. separate these two strands. But the key to success here is, what colour am I going to use to draw the new strand? Is it going to be the heavy isotope, or is it going to be the light isotope? Well, they're cultured with the light isotope, so we've only got light isotope available, so we're going to make them all with the light isotope. And so we've got two intermediate ones, and we've got two lighter isotopes. And then when we move down, we do the same thing again, and I'll do that in the model answer video, and we're still only using this yellow pen. And we just need to explain the peaks, say what peaks we've got, explain it. In generation 3, say which peaks, but you notice that one of the peaks is higher than the other rather than both being the same height. You'll notice that and relate that to this situation over here. And that's 10 marks. Reasonably straightforward if you know that experiment.